Hello there. Welcome to this website telling you all you need to know about the fibroblast growth factor receptor 2 protein and its role in Crouzon syndrome. Whether you have a family member or a friend affected by the disease or just want to learn more, you have come to the right place. I'm going to take you briefly through this diagram so that while you're exploring the rest of the website, you'll have a bit of a background when looking closer at the complications that cause and arise out of Crouzon syndrome. Let's begin. In our bodies, we have 23 pairs of chromosomes. These are parts of our cells which you can think of as where all the information is stored to make us who we are physically. Here we have a depiction of chromosome 10. This is where the FGFR2 genetic information is stored, up here. You don't have to worry about the Q26.13 part. This is just a more precise way to locate exactly where it is along the chromosome. In Crouzon syndrome, FGFR2 information is changed from what is normal. It is mutated. Some people inherit this change, and other times it can occur spontaneously. If we go down to the next part of the diagram, you can see a part labeled mRNA. This stands for messenger ribonucleic acid. Our cells have mechanisms to copy what is set out on the chromosome so that we can send it to the factories of our cells to produce the protein. This is what the messenger RNA does. These boxes here indicate the protein coding regions. Long story short, some regions in our DNA are not used for everyday instruction manuals, so our bodies need to just select the regions that do code for instructions, and these are called exons. The FGFR2 gene has 24 exons. Here the mutations I've decided to show you are common, and all of them come from the same coding region, exon 10. There are many other mutations in this same region, indicating that this is a hotspot for Crouzon syndrome causing mutations, and this is understandable considering it is an area which is important for how FGFR2 becomes activated. The mutations show the DNA base pair that has changed, and the subsequent impact it has had on the amino acid expressed. This is important because it can change the way the FGFR2 protein is folded and shaped. Proper folding ensures proper function, as proteins interact with other things in our bodies. So here you can see that the guanine is mutated to an adenine, and this causes downstream effects where if it was just a guanine it would be a cysteine, but now it is turned into the amino acid tyrosine, and this will change the folding. The numbers here are just the positions along the base pair and the amino acid sequence respectively, where it is located. It's the same for these other mutations. I will now direct your attention to this image here. This isn't a full picture of the FGFR2 protein, but it is only showing the section of the protein that is hanging outside the cell. You can imagine this, what I'm drawing here, as a cell membrane. As you can probably tell, this particular image is showing identical components of the FGFR2 protein and the FGF binding factor to them on the left and the right. Here are the immunoglobulin-like domains. There is Ig domain 2 and 3 are being shown. These are the parts of the protein that interacts with the external environment and can recognize when the protein needs to start signaling to the cell. Here are the fibroblast growth factors, and these have been used in the particular structure determining experiment to show how and where they bind to these FGFR2 domains. This is of interest because it shows where the FGFs bind to the domains of the FGFR2 and also shows that it brings these two domains together, and it forms something that is called a homodimer. You can explore what that means later on in other parts of the website, but it is essential for the activation of the protein and cell signaling. You can see the spheres here and here, and these indicate disulfide bonds. I've highlighted in red the mutations, and you can see that these mutations are very near the disulfide bond, and even right on the disulfide bond, which will be important to keep in mind as you explore why these mutations affect function. The next stage I've shown shows how normal FGFR2 protein and FGF binding to it will cause the dimerization and activation of cell signaling. However, the mutant FGFR2 can signal even when there is no FGFs here. And this is dangerous because it means it is unregulated. This has to do with what we said earlier. Because the disulfide bonds are interrupted in the mutations above, the disulfide bonds would form or not form where they normally would or wouldn't. These unusual disulfide interactions can cause dimerization independent of the fibroblast growth factor binding, and thus continual activation of the FGFR2 protein. In Crouzon syndrome, this unregulated signaling can lead to early differentiation of cells, and osteogenesis can begin early. And this makes bone in the skull and is important especially during development. So this means that the sutures in skull bones of the child, as you can see here as one suture and all these other sutures, they will fuse too early. You can see that this will cause an unusual shape, as can be seen in the photo here, and this will potentially affect the growth of other facial features like the eyes and the ears and the airways. Thank you for watching. Please explore the remainder of the website for more detailed information.